Hello students, it's Professor Brinton here, and I'm going to be giving you some help studying for your upcoming exam as well as the final. Some of this is based on the cards that a handful of your classmates submitted during the review session today. So first off, I had a request for a bit of a black box. Some final exam crashes, basically topics that often cause students problems on the final exam. So let's take a look at that list first, and then what I'll be doing is I'm starting up a series of videos called the Note Card Review, and those note card topics that you submit to me are going to be turned into very short 1 to 2 minute videos on that topic. Here's our list of frequently missed topics on last semester's final exam in particular. There's no guarantee that these exact topics are on your final exam, but obviously there are a lot of major issues, or a lot of major topics on here, so it's pretty likely. First off, basic types of bonds. As far as polar versus nonpolar, one of the note card reviews will be dedicated to that. And in particular, types of bonds in each biomolecule. So knowing, for example, that the glycosidic linkages are for carbohydrates and that peptide bonds are for proteins. That ties into polymers, how you break bonds, form bonds, pH. Lots of polarity and electronegativity questions are frequently missed. And then moving on from there, things like isomers, carbon skeletons, which there's actually already a video up for that if you want to go ahead and review carbon skeletons and how that works. Thermodynamics, unsurprisingly, is always a challenging topic, so connecting thermodynamics and enzymes tends to be even more challenging. That's where the transition state comes in. I'm definitely planning on doing a note card review on that. One of your classmates definitely requested a cell membrane transport review, and there are definitely connections there to tonicity and to electrochemical gradients, which are essential to your understanding of metabolism and how some of the very important structures, such as your mitochondria, actually function, particularly with ATP generation. Gene expression is always a topic that I'm surprised at how many students get hung up on this. Even simply on the number of nucleotides versus codons versus amino acids. Short version, 1-to-1 with nucleotides between DNA and RNA. You've got 3-to-1 between nucleotides in RNA or DNA and your actual amino acids when you get to the end of the process. That's all linked to the fact that one codon is one amino acid. However, a codon is 3 nucleotides. The replication fork and all of its associated enzymes is simply a lot of information, so occasionally students will mix things up. Not a problem there, we can review that. Chromatin versus condensed chromosomes is an essential portion of reviewing and understanding mitosis and meiosis at each particular step. And then along those same lines, both the purpose of mitosis and meiosis, homologous chromosomes versus sister and non-sister chromatids, which we actually talked about in the review today too, as well as ploidy, understanding the connection to number of copies of a particular chromosome in an organism's cells. Genotype versus phenotype versus the environment is a topic that we haven't talked about extensively, but in particular understanding that genotype is the genes, phenotype is any physical characteristic. Anything that you actually have is the phenotype. So for example, the shape of your ATP synthase structures in your cells, in your mitochondria, that is a phenotype. Even though that's something very specific and it sounds fancy and scientific, it's an actual thing. So it's your phenotype. It's an expression, a result. The environment, of course, modifies both your phenotype and can potentially cause mutations in your genotype, so there are some strong connections there too. Last but far from least, I frequently see students mix up a variety of concepts tied to karyotypes and number of chromosomes in an organism. So I definitely have a note card review planned for that one, but definitely a good idea to review for the final exam next week.